Hello, everyone. My name is Elaine Hernandez Gonzalez, and today I'll be talking about my project building a B-cell differentiation model for X-linked agammaglobulinemia, or XLA, using pluripotent stem cells. X-linked agammaglobulinemia is a primary immunodeficiency caused by mutations in the Bruton's tyrosine kinase gene, or BTK. Here we see that when a patient has a mutation in BTK, they're not able to develop the pre-B cell stage because the pre-BCR or pre-B cell receptor is not developed due to the mutations in BTK. What this means clinically is that patients are not able to develop antibodies and they cannot fight off infection. In response to that, the Porteous lab has been trying to develop a gene targeting correction for XLA. However, there's not a robust B-cell differentiation model to test this correction in vitro. Therefore, because studies have shown that transcription factor mediated differentiation has been successful in other cell types, this project suggests that it may be possible to achieve similar results using B-cell specific transcription factors instead, in order to achieve B cells. This project was divided into 4 parts. First, we wanted to establish the efficiency of previously established differentiation methods, such as the cytokine-based B-cell differentiation. Then we wanted to develop XLA mock cell lines. Then we wanted to also develop another type of cell line, which would be a reporter GFP cell line. And finally, we wanted to design the transgenes containing the key transcription factors that would drive B-cell differentiation, as I mentioned before. First, here we see the efficiency of pluripotent stem cells to differentiate into B cells. We have CD34 cells as the first marker, and this progresses in this direction until we finally reach the CD19 and CD20 markers, which are B-cell specific markers. So here we see that cells are almost completely committed to not being CD34 cells, and they move on to further stages in development. However, when they're closer to B-cell stages, they lose this ability to completely commit to B cells. So there is a lot of cellular heterogeneity, meaning that these cells are, this differentiation system is not very efficient in pluripotent stem cells. Afterwards, we established knockouts on different pluripotent stem cell lines, embryonic H1s, and also induced pluripotent stem cells 1 and 2. And these are all mill cell lines. And here we see that they're just, by using 3 guides, we're able to create a very sturdy knockout of these on each cell type. Then we moved on to developing CD20 reporter gene constructs. CD20 is encoded by the gene MS4A1, and this is a B-cell specific marker that is present after the pre-BCR stage, which is what we're trying to achieve. Here we use 2 guides, one on the 5' end, one on the 3' end, and we develop these constructs on both sides of the gene just to determine their efficiency. We tested on HSPCs and B cells, and we saw low integration rates as well as low GFP. However, we don't see that CD20 construct 2 impacts a lot of the marker expression compared to wild type conditions. Regardless of this, we want to repeat the targeting experiments just because there were limitations in these experiments, and the viral load could be improved as well as the enhancement of the integration. Finally, we designed our EBF1 impacts 5 gene constructs. These 2 are transcription factors that are B-cell specific, and here we designed them on 3 different constructs separated and together with their selection markers. And most importantly, or most interestingly, these dehydrofolate reductase parts, which are destabilization domains that basically cause the induction of the transcription factors themselves, because these, once you add a small molecule, these constructs can be stabilized, and otherwise they're not stable, and therefore they cannot increase the differentiation uncontrollably. Therefore, to conclude, we were able to test previously established B-cell differentiation systems. We saw that BTK knockouts and the XLA mock cell lines are possible to create. Afterwards, we began to study the different CD20 GFP constructs, but more testing is necessary to determine which one is the best one. And finally, we want to move on with our transcription factor constructs by actually targeting cells and seeing how efficient they are at driving B-cell differentiation. Thank you for listening.